Rahman Rahim. Now let's see what happens after the formation of the neural tube uh, in the development of the nervous system. Well, this figure over here on the left side shows that the neural tube has completely formed, and the point that this figure makes is that the rostral part of the neural tube, it develops much more than the caudal part, and it forms three primary and it undergoes cephalization, and it forms three primary brain vesicles: the forebrain, the midbrain, and the hindbrain. And then those three primary brain vesicles are going to give rise to five secondary brain vesicles. Five secondary brain vesicles. And the entire central nervous system will be developed from these secondary brain vesicles, except the spinal cord. And this is another figure that illustrates the forebrain, the midbrain, and the hindbrain. The forebrain, these are the three primary brain vesicles. The forebrain is known as the prosencephalon, the midbrain is known as the mesencephalon, and the hindbrain is known as the rhombencephalon. And you can see that this is how the rostral part of this of the neural tube has developed more than the caudal part, and it forms three major primary brain vesicles: the prosencephalon, which is the forebrain; the mesencephalon, which is the midbrain; and the rhombencephalon, which is the hindbrain. And from these three primary brain vesicles, you get five secondary brain vesicles. So let's see what structures are derived from each of the five secondary brain vesicles, because each of the secondary brain vesicle will give rise to a specific part of the central nervous system. For example, the telencephalon. Basically what you see over here, the forebrain divides into two structures, two secondary brain vesicles. One is the telencephalon and one is the diencephalon. And from the telencephalon, the cerebral hemispheres, they develop, and also the basal ganglia. From the diencephalon, the thalamus develops. The midbrain gives rise to only one, remains the same. It's only the secondary brain vesicle is formed, which is the mesencephalon, and it forms the midbrain. The hindbrain divides into two secondary brain vesicles, the metencephalon and the myelencephalon. The metencephalon forms the pons and the cerebellum, and the myelencephalon forms the medulla. And you can see in this figure that the three primary brain vesicles have divided into the secondary five secondary vesicles, and the forebrain has divided into two parts. The forebrain is known as the prosencephalon and divides into the telencephalon and the diencephalon. The telencephalon gives rise to the cerebral hemispheres, as you can see, and the diencephalon will give rise to the thalamus. The mesencephalon does not divide; it remains the same. It remains one. This is the second primary brain vesicle, and it develops into the midbrain. And the hindbrain divides into two secondary brain vesicles, the metencephalon and the myelencephalon. From the metencephalon, you get the pons and the cerebellum, and from the myelencephalon, you get the medulla oblongata. Another thing to note is that the neural tube, during its development, it envelops it's hollow from the inside, so it envelops a hollow central canal, which later develops into the ventricles of the brain. And each of the secondary brain vesicles, they envelope particular ventricle. For example, the cerebral hemispheres, each one of them contain a lateral ventricle. So there are two lateral ventricles, one in each cerebral hemisphere. And the thalamus, which is part of the diencephalon, it envelops the third ventricle, and the pons and the cerebellum and the medulla, the upper part of the medulla, the pons and the cerebellum, they envelop the fourth ventricle. Now to note is that the midbrain is the only secondary brain vesicle which does not envelope a ventricle. Instead, it envelopes the central canal remnant, which is known as the cerebral aqueduct. And another central canal remnant is enveloped by the spinal cord. So now let's see which of the secondary brain vesicles give rise to which structure in the adult nervous system. The telencephalic structures, as we said, that from the telencephalon, the cerebral hemispheres they develop, and most of the basal ganglia, they are subtelencephalic remnants, and they enclose the lateral ventricles. And the diencephalon, there are four structures that develop from the diencephalon, all ending in the word thalamus. The main thalamus, which is the relay station and it's a collection of sensory and motor relay nuclei, and whichever neural system wants to influence the cerebral cortex must relay inside the thalamus. Very important structure. The hypothalamus, it controls the autonomic function and the endocrine function. The subthalamus is a part of the basal ganglia, and the epithalamus is another fancy name, a word for pineal gland. And the retina and the optic nerve are also diencephalic structures. The optic nerve, it grows out of the diencephalon and the retina as well. And the diencephalon envelops the third ventricle. The mesencephalon is the midbrain. It is the secondary brain vesicle which does not divide into any other remnants, and it is surrounded by it surrounds the cerebral aqueduct. The metencephalon, this is the hindbrain, develops into two vesicles. The third primary brain vesicle divides into two secondary brain vesicles. The metencephalon, which develops into the pons and the cerebellum, and they enclose the fourth ventricle. And the myelencephalon forms the medulla. And what's not correct in this chart, the spinal cord does not develop from any of the secondary brain vesicles. The spinal cord remains the original remnant of the neural tube. The medulla encloses the fourth ventricle. So there's a little correction on this chart. This chart is not completely correct. Sorry about that. This is the correct chart. The metencephalon and the myelencephalon, these are the two secondary brain vesicles that are derived from the third primary brain vesicle. The third primary brain vesicle divides into two vesicles, secondary brain vesicles, the metencephalon, from which the pons and the cerebellum develop, and the myelencephalon, from which the medulla develops, medulla oblongata, and the cavity that they envelope is the fourth ventricle. And the spinal cord does not develop from any secondary brain vesicles. It is a derivative of the original neural tube, and it envelops the central canal. So this table summarizes what we've talked about so far. The entire nervous system develops from the ectoderm, which develops the notochord from the mesoderm, induces the part of the ectoderm just lying above it to form the neural plate. The neural plate forms the neuroectoderm, and the neural ectoderm then divides into the neural tube and the neural crest cells, which are outside the tube, and then there are structures which develop from the tube, which is the entire central nervous system. The retina and the optic nerve, they are outgrowths of the diencephalon. The pineal gland develops from the neural tube, the neural tube derivative, although it's an endocrine gland. The neurohypophysis is the posterior pituitary. The anterior pituitary is formed from the oral ectoderm of the Rathke's pouch, and the astrocytes are the glial cells which form the scaffolding of the connective tissue of the central nervous system. And the oligodendrocytes, they form the myelin sheath of all the axons in the central nervous system. If you come to the neural crests, cells which have migrated into the body peripherally, the adrenal medulla is derived from the neural crest cells which have migrated, and they act as a sympathetic ganglion. Instead of sympathetic ganglion, the adrenal medulla, since it is derived from the neural crest, it performs the function of sympathetic ganglion. And the preganglionic fibers, they do not synapse, they come and they innervate the adrenal medulla, and when they are stimulated, the adrenal medulla releases epinephrine or epinephrine inside the blood. So instead of the sympathetic ganglion, the adrenal medulla is there because it is derived from the neural crest cells. Then all the ganglia in the peripheral nervous system are derived from the neural crest cells. And all the primary sensory neurons which collect information from the environment and bring it to the central nervous system. And all the autonomic postganglionic neurons, they are derived from the neural crest cells. Then the melanocytes in the skin are derived from the neural crest cells. The Schwann cells, they are the myelin producing cells of the peripheral nervous system. They are derived from the neural crest cells. Then the meninges, the pia and arachnoid, are derived from the neural crest cells. The dura is a derivative of the mesoderm. And all the connective tissue and cartilage of the rest of the body is a mesoderm derivative, except the pharyngeal arch cartilage. The pharyngeal arch cartilage is derived from the neural crest cells. The odontoblasts are the cells which form the teeth, the dentine of the teeth. And then the parafollicular cells, the calcitonin producing cells in the thyroid gland, they are derivatives of the neural crest cell. The aorticopulmonary septum and the endocardial cushions, they are all derived from the neural crest cells.